Hello, everyone. You are welcome to today's edition, which is the sixth edition of Install African online sensitization program for strengthening fundamental research capacity in Drosophila in Africa. My name is Rashid Omotayo Adeyemo, and I'll be standing in for Madam Aksa Iguzaneza, who is unavoidably absent. As you know, she's our usual moderator. But nevertheless, it promises to be educating and fascinating as we have a big fish in the house as she'll be taking us through our journey in the world of science. Without further ado, um, I don't know if Mr. Emmanuel Munezero is around to take us through the profile of our guest lecturer. Mr. Emmanuel, okay, you are welcome, Prof. Ladies and gentlemen, may I permit me to go through the profile of Professor Dolores Martin Bermudo. Dr. Dolores Martin Bermudo, please pardon me if I'm not pronouncing your name very well, please. Dr. Dolores Martin Bermudo is a distinguished researcher specializing in fundamental and cell biology. She holds the position of Investigador Científico at CSIC, based at Camp Pablo de Olavide in Seville, Spain, with a master's and PhD from Universidad Autónoma de Madrid. Her extensive career spans various prestigious institutions, including a Royal Society University Research Fellowship at the University of Cambridge. Dr. Martin Bermuda's research centers on Drosophila melanogaster, focusing on developmental and cell biology. Over the past decade, she has supervised 12 PhD theses, contributing significantly to scientific knowledge. As a corresponding author in 34 out of 58 publications, her work boasts of an H-index of 24. Currently, investigating epithelial morphogenesis and cell migration, Dr. Martin Bermuda's group identifies new genes regulating individual and collective cell migration, studies invasive behavior of epithelial cells, and analyze the role of cellular tension during morphogenesis. Noteworthy recent publications include studies on integrins, notch pathway modulation, and actomyosin dynamics. In the realm of scientific funding, Dr. Martin Bermuda secured several grants, notably from the Ministerio de Economía, Competitividad, and Ministerio de Ciencia e Innovación. Recognition include the EMBO Young Investigator Prize in 2002, EMBO membership in 2020, and Suffrage Science Award in 2022. Beyond her impactful research, Dr. Martin Bermudo actively contributes to scientific community service, serving on grant review committees as an ad hoc reviewer for esteemed journals. Our commitment extends to monitoring, having supervised 9 PhD students, and engaging in various teaching roles. Dedicated to science outreach, Dr. Martin Bermudo co-founded Droso Africa, a charity promoting Drosophila as a model system for biomedical research in Africa. Our participation in research, events, and advocacy for women in science further exemplify our commitment to advancing scientific knowledge and fostering inclusivity in the field. Ladies and gentlemen, permit me to welcome Professor Martin Bermudo as she takes us through her journey in science. Professor, please, the floor is yours.
Okay, let me share. Uh, can you see my screen?
Partially.
we can see the screen.
Can you see my screen or?
Yes, we can now.
Okay, let me see. Okay, thank you very much for this very nice introduction. It's a real pleasure for me to be here with you guys. And what I try to do today is to sort of get you through why Drosophila is such an important model to do research in human diseases. Okay, so you see my presentation now?
Yes,
we
can see.
Okay, great. So
you can try to make it full screen.
Sorry?
You can try to make it the full screen.
A full screen. Let me see.
Presentation mode.
Yeah, no, I am in presentation mode, but let me try to do something else. Hold on a second. I am in presentation mode, but hold on. I'm going to try to make it like a full, okay? View, slideshow. No, in view. I think that I should go to slideshow, play from start, present to view. Now? Is it full now?
Nothing has really changed.
No, I am in full screen in my screen.
I
Yes,
think
it
it's
is
coming
full.
up gradually.
Sorry?
It's coming up gradually, Prof.
Okay. Okay, so I'm going to do like a brief summary of my career, no, that has already been done very nicely. So I studied the nervous system. I have been working with Drosophila for nearly 40 years now. So I started to work trying to understand the formation of the nervous system in Drosophila during my PhD. Then as a postdoc, I went to the University of Cambridge in England, and I studied organogenesis in Drosophila. Then I went back to Spain and got a permanent position where I focused my research in cell migration and invasion. And since then, I have also widened my studies to epithelial morphogenesis. All of this using Drosophila as model system. Can you see my slides? Hello?
Yes, we can see.
Okay,
Yes,
great. Okay. So why do we think that Drosophila is a good model to understand human diseases? Okay, so this is the fly, the Drosophila fly, which is a very little fly that you can find in the fruit. And this is a closer view of Drosophila that many of you has to be familiar with it. Okay, and as you will know, Drosophila, as any living organism, has all the tissues and organs as humans. So they have a respiratory system, they have an intestine, they have muscles, they have a circulation system, they have a brain, and also they have a very similar body organization plan. So they have the head at the top, then they have the thorax and the abdomen, and then they have legs. So the structure of the body and the organization of the body is also very similar to humans. Okay? So not only that, but Drosophila is very easy to culture and maintain in a lab. All you need is a plastic small tube with food so that the fly can lay eggs. And then this food is just made of flour, basically. And then you can keep and store lots of different type of Drosophila, mutants, wild type, very easily in an incubator in trays, as the picture you see here. And they are very easy to manipulate under a normal dissecting microscope. All you need to do is to be able to manipulate them, to sleep them, and you can use CO2, or you can also use like a ether to sleep them. So it's very easy to culture, maintain, and work with. In addition, Drosophila has a very short life cycle. So it takes like 10 days at 25 from the embryo, from when the embryo is laid until you get another fly, another adult. Okay, so the embryo develop into 3 initial larvae stages, then they go into prepupa, they pupate, and then the adult comes out. So it's only 10 days. In contrast to, for example, models such as the mice that you have to wait a long time to be able to get the progeny. In addition, and more importantly, Drosophila has only 4 chromosomes, okay, instead of the 23 of the human. And it's very easy to generate mutants. And you see an example here where this is a mutant, a Drosophila mutant, that the eye is white instead of red. But there's lots of new techniques like the CRISPR technique that allows us to generate a mutant in any gene of interest. Okay. Also, there is powerful techniques, and this is just an example of a tomography done in an adult fly. So you can see all the organs. You can see the ovaries here. You can see the muscles. You can see the eye, the brain. You can see the intestine here in yellow, and you can see the excretory system. Then you can also see, as I mentioned before, the ovaries. Then you can see muscles that allow you to fly, other muscles that allow you to walk, so that if you generate mutants in a specific, for example, disease, you can analyze what's wrong with the body, with the formation of the body. So this is really good to study developmental biology, for example, or diseases. And then you can also look in vivo. So this is an example of a Drosophila embryo that we do in the lab, and in red, you have hemocytes. We have generated the wound, and then you can watch in vivo how the hemocytes close the wound. So there's lots of in vivo analysis that you can do. I can repeat the movie. So red are macrophages. In green is the epidermis, the ectoderm. We generated the wound with a laser, and all the macrophages come to close the wound. So we can study wound healing, for example, but we could study invasion. We could study many things in vivo. Okay? So the genome of Drosophila is totally sequenced, okay? And there's lots of similarity with the human. So this is a comparison between the human and Drosophila melanogaster. And in blue is the catalytic and regulatory domains of proteins. And in yellow is the association of the genes. So if you look at it like a very quick view, you see that nearly all the bases are somehow conserved, okay? And importantly, the catalytic domains and the regulatory domains are very conserved. So the proteins can do very similar things in humans. In fact, more importantly, from the point of view of using Drosophila as a model system to study diseases, out of the 929 genes linked to human diseases, okay, 700 are very well conserved in Drosophila. In fact, there is a web page which is called Homophila, where they associate the genes that, when mutated, causes a disease in human and found a homologue in Drosophila. So if you are interested in a disease, let's say Parkinson's disease, where several genes have already been mapped that, when mutated in human, causes the Parkinson's disease, you can go to the fly and search which are conserved in the fly. So, for example, PINK1 is tau. So, for example, Parkinson 2 is Parkinson. So then you can choose a disease that you would like to understand, and then see in this web page whether there is a homologue in Drosophila, and then try to use Drosophila to understand the gene, okay, the human. So you inquire a neuropathy, you do the research, and then you can find the gene. As an example, there is a gene, PAX6, which in human, when mutated, you get lots of areas of the eye which is missing, and that causes blindness in human. In the mouse, that looks like there is no eye, a mutant in the same gene in the mouse, and a mutant in the same gene in the fly causes a very reduction of the eye, similar to what happened in humans. So now we can try to understand how PAX6 regulates the morphogenesis of the eye using Drosophila. Another example is a mutation in a gene which is called HIPPO, which is a whole signaling pathway, which when mutated, for example, in the kidney of a human, generated tumors in the kidney, in the brain, and many other tissues. The same gene is conserved in Drosophila, and when you mutate it in the brain of Drosophila, you get a tumor in the brain. So now we can use Drosophila to try to understand why mutations in the hippo pathway generate tumors. Okay? So knowing that, there are many thousands of papers now out where they have demonstrated the role of Drosophila in trying to understand human diseases in several types of diseases, like neurodegenerative diseases, muscular dystrophy, brain diseases, tumorigenesis, behavior, etc. I'm going to give you some examples, okay? So, for example, in cancer. So there is a gene called LAX that when mutated generates this tumor. And then that another gene, which is known that causes glioblastoma, when mutated in the fly, it can generate a glioblastoma in the brain of the fly. And now you can use this to try to understand why mutations in this gene generate glioblastomas. Okay, this is the same gene mutated in the fly. In fact, sometimes what we do is that we generate a fly that carries a mutation of a human gene, and we put it in the fly and we try to understand. Okay? In addition, we can, for example, take tumor cells from this brain, implant them, which are labeled with fluorescence, implant them in the abdomen of a fly. This is the original site of implementation, and see how the tumor grows, and then colonize the entire fly. So we can study metastasis as well. And this is another case where there was a tumor in the eye, and then you can see tumor tissue of the eye in other parts of the body. And we can try to understand how metastasis takes place, okay? This is another example where we can study muscular dystrophy. This is a muscle of a Drosophila fly, an adult fly, and you can see it has all the set bands similar to the human muscles. And there is a mutation in humans that cause aberrant muscle contraction, and this is also similar mimic in the fly. So this is a muscle of the fly that carries these mutations, and now the set bands are disrupted, and these flies cannot move properly. Okay, so and there is an easy test to look for muscular dystrophy. For example, these flies are normal flies, and you just see how easy they climb up because they have geotaxis. Okay, so they go to the light and they go always up. If you generate mutants that affect muscles, like the one I showed you before, the flight takes much longer and sometimes cannot even get to the top because these flies have problem with muscles. Okay. Another way to analyze the behavior of the muscles is that in the adult, because we can generate mutants in the adult, and then this fly, as you see, cannot move properly. So see, this is a fly that carries a mutation in a gene that, when mutated, causes muscular dystrophy. Okay, you can see it here again. The fly cannot really walk, and if you put it upside down, you see that it has lots of problems of walking. Very similar to the phenotype that we see in humans. Another thing that you can do is study neurodegenerative diseases, and I'll give you some examples like Alzheimer's, Parkinson, Huntington, ataxia spinocerebral, lateral sclerosis. Diseases are hereditary and affect more than 106 million people in the world, which is like with an incidence of over 50% when you are over 60, okay? So this is a cartoon showing you the size of a human brain, and here is the size of a Drosophila brain, okay, for you to have an idea. So while the human brain has 86 billion neurons, the Drosophila brain has only 135,000 neurons. And now studies of many labs have analyzed all the neurons, and then we have markers for all the neurons present in the Drosophila fly. So these are the circuit of some of them. You can follow the connections. Some of them connect contralaterally, some of them connect ipsilaterally, and then you can follow the individual neurons. And you can even mutate individual neurons and see what happened to the whole brain, the function of the brain, behavior, etc. okay? Here with tomography, you can see the bodies of the neuron and how they connect to all the different places. And all this now is really well known. The arborization, the trajectory, everything is known, okay? And now you can even analyze behavior. For example, let's say that you know that this neuron is a dopaminergic neuron that has a mutation in human that impairs locomotion. So you can now mutate this neuron, and then we can use artificial intelligence to follow the behavior. And this is just an example, okay? So here, by artificial intelligence, you can follow all the movements of all the legs of the different flies, a normal fly, and a mutant fly, and another mutant fly, and you can measure lots of things. You can measure the movement of the legs, you can measure the movement of the body as they walk, okay? So for example, here we are measuring how the body changes, and then you can, by artificial intelligence, it will draw you all the movements along a period. And then you can mutate individual neurons, or you can express a mutant form of a gene that affects locomotion in humans, and then you can see how that affects the movement of the fly. So we use artificial intelligence in flies to analyze cellular behavior, okay? We can go even farther. And I don't know whether you can see this here, but this is a tiny fly that is located here. And then what this group has done in Portugal is that they open a little window in the brain of the fly, like this, okay? So you generate a little window, the fly is still alive, and then you have access to the brain. And then with the objective, you can follow the behavior of the brain and the activity of the different neurons. For example, here, what they are studying is how the fly behaves anti-certain stimuli, okay? So they offer the fly an odor or another hormone, a sexual hormone or whatever, and they see what neurons are highlighted, what neurons are active. So for example, here, they look at the activity, and then when they receive the stimuli, they see that there is this type of neurons that then become active. So then they are studying what are the set of neurons that are activated upon different stimuli. And then you can do this. This is a wild type flies with different type of stimuli. The previous one just, activated neurons that cross. This one activated this neuron. This stimuli activates now this pair of neurons, as you can see here. And then you can expose the fly to different stimuli and then see which are the neurons that are activating, to try to understand what neurons control different type of behavior, okay, at any level in the fly. And then we can extrapolate that to human. Another thing that we can do is to use flies to look for components, to look for compounds, drug screen, to analyze what drugs will, for example, improve a behavior, a tumor, or any disease that you want to study. So, for example, there are people who are studying who are trying to isolate compounds that will prevent the growth of a tumor generated in the abdomen, like the one I showed you before. And the way you do it is that you put the flies with these tumors in little wells, and then you provide, and each well has a different compound, so you can analyze thousands of compounds. And again, using artificial intelligence and sophisticated microscope, you can see in which of these well this tumor is reduced with time, or this tumor is increased, or this tumor disappear. And then you can choose which well, which compound is acting as a drug to, for example, block tumorigenesis, or you can also do it for behavior, you can do it for many things. So Drosophila can be used for drug discovery, okay? Now I'm going to change gear to behavior and to show you how Drosophila can be used to analyze behavior, okay? So there is a group here in Germany that study Drosophila to analyze ethanol consumption. That you may think, how are you going to use Drosophila, okay, to study ethanol behavior, like if you get drunk or not, and whether that is genetically controlled. So what they did, the advantages of using Drosophila is that you have a control over the history of alcohol consumption, okay? You also can use the right amount of ethanol that you want and analyze different behavior like locomotor impairment, postural control, sedation, immobility. So as you see, the fly goes acquire an intoxicated phenotype, which is remarkably similar to the one that human expose when they have alcoholic intoxication. And then you also have control over the genetic background, and you can do screens to try to isolate genes that control the behavior upon alcohol consumption. So an example is an assay, which is called the biometer. And what they do is that they throw the flies through this tube, and in this tube there are meshes all around at different levels. And if the fly is alive, then is asleep, the fly is awake and hasn't consumed alcohol, the fly will go through this and then will find the meshes and will be able to get down easily, okay? However, as they lose postural control after alcohol consumption, they take much longer to get down. So they can measure the time the fly takes to go through the column, indicated whether the fly is drunk or not, because they can produce alcohol through this tube, okay? Oops. Another thing they can do is that analyze behavior, okay? And they do that by now introducing ethanol in this plate, which is closed, and then with a digital camera, they can follow the behavior and the movement of the individual flies. And then they can generate mutants randomly and select those mutants in which the behavior is different. So for example, they have isolated genes that control alcohol dehydrogenase. They have annullated genes, which is a neuropeptide, which is produced in the brain and controls the ethanol behavior. The gene was called amnesia. They can also find genes regulated dopamine, which respond to alcohol consumption. And as I say, in the first exposure to alcohol, the flies just start to move randomly, and then after a while, they fall asleep. One thing that this group demonstrated is that after a second exposure, the fly becomes a little bit resistant to alcohol, and now it takes much longer to go to sleep. So the first exposure, after 12 minutes, the fly cannot move because they're totally drunk. After the second exposure, the flies have become a little bit resistant to alcohol, and then 12 minutes later, there is still lots of flies. I wish that was the case in humans. Only second exposure to alcohol, you are now resistant somehow, but this is not the case. So then they can analyze what did the response is altered, and they have identified lots of different genes. Another behavior that they can analyze is sexual behavior. And this is just a movie of a spider that then display this beautiful pattern here to attract a female. Because the sexual behavior is very important in all the species, because the main purpose of a species on Earth is to reproduce itself and be permanent on Earth, okay? So reproduction is one of the key reasons, of the key function of a species, as a species. From evolutionary point of view, all species needs to reproduce. So, and they have devised lots of really important and key ways of controlling the sexual behavior so that they are successful. This is the case. Making noise also with the legs until they convince the female that they have to copulate. Okay, so this is a very stereotypic behavior. And then with the flies, you can analyze how this behavior is controlled, because this behavior has been shown to be controlled genetically. So what some people have discovered is that there is a gene which is expressed in males, which is called sex lethal. It's not expressed, this is the brain of a male. And then in green is the expression of a gene which is absent in females. And these genes confers the males the ability to behave the way I just showed you. And they have shown that because if you mutate this gene in males, the males start to chase each other and they ignore a female which is in the plate. So they start to chase other males, and it doesn't matter, the female is here and they ignore totally the female. Okay, they just pass by, they ignore her, and she tries to chase her, but they ignore her. Okay. On the other hand, if you express the gene in the female, that is enough in the brain of a female, that is enough to trigger a behavior in the female which is similar to the male. So now this is a plate where all of the flies here are females, and as you see, now they are behaving exactly like males. They start to move the wing perpendicular to the body and make a chain, you know, that they just follow the male in front. So they just make this change, behaving as a male. So this is just the expression of a gene in a subset of neuron in the brain. And by that, they can analyze lots of different sexual behavior. Okay? So now they can also analyze mating behavior, and they have started to identify neurons that express specific genes in the brain that analyze the mating behavior. And this is a video showing you how I just showed you that the males follow the females, but also the female has to let the male to copulate, has to let the male to mate with them. So it's not just that the male is the one controlling the sex mating behavior, it's also the female. So the female produces a pheromone that attracts the male. So in Drosophila, if the female has been mated, she does not allow other males to mate with her. And I can show you the behavior here. So you see this female is repelling the male, so the male cannot mate. Then she produces a pheromone, and then she will let sooner or later the male to mate. Okay? There, at that moment. So then they are now trying to understand what are the hormones that the male produce, so that the female produce, that they attract males. How is this controlled at the level of the brain? And this is really important because they can also be used in agriculture when you want to stop, like, you know, insects from mating, and then you can sort of block a plague of insects that you don't want to have in your crops, for example, no? There is also aggressive behavior, and I can show you here how two males are fighting with each other. And this is also controlled at the level of the brain, obviously. And now they are trying to analyze what genes control this behavior. And then you can do that by generating mutants randomly, and then isolate those mutants in which this behavior is stronger or this behavior is weaker, so that you can also try to go into the molecular mechanisms regulating aggressive behavior. Okay? So with that, I hope I have convinced you that the Drosophila fly is a very powerful system to analyze human diseases, human behavior, to analyzing drug screen. So it has lots of potential for biomedical research, being a model which is very cheap to maintain, that basically anyone can have it in the lab, that there are very little constraints to use the model, because, you know, like, for example, if you work with mice, you have to have an animal house, there's lots of rules, but with Drosophila, there are very little rules, just to work with it, so that nearly every lab in the world can use Drosophila to try to address a question. So actually, using Drosophila, you can address any question that you can think of. So the limit to research is basically your curiosity, your ideas, and your willingness to do research. Okay. So having this in mind, we have decided, Marta, Isabel Palacios, and myself,
maintain and to manipulate to generate to simulate a lot of diseases because of the similarities between human beings and Drosophila. She did talk about the fact that Drosophila has 4 chromosomes, which are very homologous and similar to the chromosomes we have in humans. She talked about the fact that they have a short lifespan, which is very easy to study a lot of diseases, particularly neurodegenerative diseases such as Parkinson, Alzheimer, lateral sclerosis, all those neurodegenerative diseases. Also, Drosophila is very useful when it comes to drug discovery. If you want to see the effect of isolated compounds, how the system will behave to a particular compound, as well as different behaviors. For example, she did talk about the alcohol-induced behavior and also the mating behavior in Drosophila. So these are some of the things she mentioned, pointing to the fact that Drosophila is a very useful tool to model a lot of things in human beings. So, ladies and gentlemen, the floor is yours. I don't know if you have questions for our professor so that she could answer those questions without wasting much of time. Let me go through the box to see if there are questions.
I can see question about the research of aging and infectious diseases in the chat.
Yes.
Okay.
Yes.
So, Rashid, thank you very much because you've done such a lovely summary of it. It's impressive. Thank you very much.
You're welcome, Prof.
So yes, I mean, aging has been extensively studied in Drosophila, basically because it's very easy to study aging because the cell cycle is so short and the fly doesn't live that long in general, okay, that you can easily isolate mutants that affect the length of the life length, the expansion of aging. And then by doing that, and what the people have done is that generate random screens so that you mutate randomly the gene, okay? And then you find flies and then you study the flies and see how long they live. And then you isolate those mutants in which the fly either live longer or the fly live shorter. Then there are ways to isolate those mutants and to identify the molecular lesion. So you can identify the gene that when mutated make the fly to live longer or shorter. And many genes have been already identified. You can also analyze how a specific disease affects the aging, the length of the life length of a person, of a fly. So for example, I think that it has been shown that diabetes affects the lifespan of a fly, okay? And many other diseases affect the lifespan of the fly. Another thing that the people are analyzing in aging with Drosophila is that how that affects fertility, how that affects for example, the immune response to an infection, how does affect the formation of a tumor, how does affect other type of behavior. So the people are now using like old flies and then try to assess generating, for example, an infection with the bacteria or something and see how the fact that you are old then affect the way you fight with that infection, okay? Because actually the fly has also been used to analyze infection diseases and how the fly respond to an infection, either a bacteria or I think that mainly bacteria or on fungi as well. And then you can analyze the behavior of the fly from the point of view of the immune system, how macrophages fight the infection, what are the resources, where the signals come from, how is the signal that you have an infection is transmitted to the brain so that the brain can then activate the immune system to fight to that infection. There's thousands of papers, or hundreds, let's say, of paper analyzing the different, the molecular mechanisms behind fighting an infection.
Thank you, Prof. Thank you very much. I hope that answers the question very well. There's another question here, Prof. The questioner is asking, can they be used for dermatotoxicity?
I'm not sure what you mean with dermatotoxicity.
Um,
What?
maybe the effect of I'm just trying to read the minds of the questioner now. Maybe the toxic effect on the skin.
Yes, well, not really on the skin, okay, because the fly doesn't have such a like something like a skin. It has an ectoderm. But so I'm not sure whether you could use how toxic a compound for a human skin is for the fly. Maybe if that produces cell death of the skin, then you can analyze whether a compound can produce cell death in the skin of the embryo, for example, or whether it can induce proliferation, or whether it can affect the way the cells attach to each other. Maybe that kind of thing you can analyze.
Thank you very much.
Because
Another
also, for
question.
example, the intestine have an ectoderm, in this case an endoderm, no, which is an epithelial sheet. And then maybe you can feed the fly with a compound and see whether the epithelial sheet of the intestine is somehow affected upon any compound. You could also
Oh,
use that, I guess.
okay. Thank you. Another question reads, can we use Drosophila melanogaster to study psychological disorders such as depression and anxiety, genetically using gene SS and SL? That's the
I think
question.
that I think that some psychologically disorders, like for example, if you are autistic, okay, that has also, I mean, no autism in on its own, no, because I don't think that it's really well known whether what are the genes that, or whether what is the genetic cause of the autism, okay. But definitely the people are studying psychological disorders related to the way you see other humans, like the way you relate to people similar to people like you, no? So because there is a very social behavior with the flies, so they normally behave like they don't become aggressive, they relate to each other. So you can try to analyze how the fly relates to each other, and that is being done.
Thank you very much. Another question reads, I need some explanation about how different strains of Drosophila melanogaster responds to treatment, especially in nutrigenomic studies.
Okay, so what the people are now that doing a lot of nutritional studies in Drosophila. Okay, so they try to understand from the point of view of the intestine, but also from the point of view of how the brain regulates, any nutritional problem. So because there is a lot of nowadays, it is known that interorgan communication between the intestine and the brain, between the muscle and the brain, is very important, okay? And many things are controlled at that level of interorgan communication. So what the people try to do for nutrigenomic studies, for example, is they try to analyze how an infection can affect for example, the composition of the bacteria in the intestine of the fly. Okay, because you can isolate the intestine of the fly, and then you can do nutrigenomic, or you can do analysis at the level of genomics of the bacteria which are inside the intestine. And then you can try to analyze whether, some people with some disease, like they have even mimic bulb disease in the fly, because sometimes there are mutations in it, and I know that they affect the way you, for example, the way the intestine treat the different nutrients. And then you can mimic that disease and then see what is the impact in the growth of the larvae, because if you don't eat properly, or you don't get your nutrient properly, then you don't grow properly. Okay, but also even at the level of the excretory system. So there are people analyzing how many times the fly go to the toilet, let's say. So they can analyze that. And they can also, by tomography, there is a group, Irene Aliaga in London, that do lots of tomography to analyze the consequences of nutrition in the growth, in the repair of the intestine, because we repair the intestine fully every. So all that can be studied in the flies.
Thank you very much. Let me combine these two questions. Can Drosophila be used for non-communicable diseases such as diabetes and heart diseases, as well as malaria infection?
Yes.
Could it be used in malaria infection studies?
Yeah, as diabetes, actually, some of the genes that are implicated in diabetes were isolated in Drosophila. Okay, like for example, Chico, which is the insulin receptor. So there's lots of groups now analyzing diabetes in Drosophila because the insulin receptor, for example, of Drosophila is very similar to the one in humans. So they are trying to understand, how diabetes is controlled. And probably there will be groups trying to analyze compounds that will help, because you can also measure the levels of glucose in the fly. You can also measure how the fat body, it is known that the fat body produces and excretes morphogens and, like compounds that are read by the brain, and then that will control the level of glucose. So if there are very, very little insulin, then the fat body will produce some of these morphogens to be able to increase the level of glucose. So there's lots of study, not only at the level of the cells that produce the insulin, okay, that there are cells that produce the insulin in Drosophila as well, but also how the brain knows that there are low levels of insulin, and how the interorgan communication, again, can compensate for low level of glucose, high levels of glucose, okay? With respect to heart diseases, Drosophila also have a heart, but it's very different from the one that exists in humans, because it's a very long structure, okay? But it also has myocytes. And then the people are trying to analyze, for example, how the extracellular matrix regulates the function of the Drosophila heart, because the Drosophila heart has also muscles surrounding it and has also a matrix that surrounds it. So there are many laminopathies that affects the matrix and therefore causes coronary diseases in humans, and that is shown that also affects the beating and the activity of the heart in Drosophila. So it has been used for that. And malaria, I have to say that I'm not sure whether malaria is specifically being studied using Drosophila. I know that many other infectious diseases, but malaria, you should look in internet and say malaria and Drosophila, maybe they're groups. Because, to study malaria, you need a very sophisticated lab. That you need, like, labs where malaria cannot go out and all that. So I'm not sure whether malaria is being studied. Maybe yes. Why not?
Thank you very much. There's a clarification on the dermatotoxicity that the questioner earlier mentioned. So the clarification is that would be to test on the thoracic region and the effect on the wings or on the larva or the guts. That's what the
Yeah,
questioner is actually asking about.
yeah. I have to say that I don't know whether Drosophila is the best system to study dermatotoxicity. Because I don't know. I mean, it's just a question to try. Maybe just look on internet. Maybe someone is doing it, and it will be a question to try to say, if I feed the fly with this that I know causes the dermatotoxicity in humans, what is the tissue that will be most affected? Ideally, one would like to have a positive control that affects a specific tissue, and then having a positive control, you say, I have a way to affect this tissue. Is this compound that produces dermatotoxicity give rise to similar effects? But I don't know whether the thorax or the wing or the larvae or the gut will be the best system. One has to try if it has not been tried, which I don't know.
Thank you, Prof. In order not to go beyond the time, there's a question here. I will just tell the person that Prof did mention about this. The person is asking about how to use Drosophila for drug discovery. She did mention about the use of Drosophila for drug discovery. Probably you can just go over the YouTube. You can play back the YouTube to understand what the prof said about this. So not to waste our time and to be time conscious, we'll just go straight to the vote of thanks to be given by Miss Blessing Pereye. Sorry if I've murdered your name.
Okay, thank you.
Yeah, yeah, thank you. You did well in pronouncing the name. Yeah, so I want to appreciate our distinguished prof, Professor Leva, for the stimulating presentation. Thank you very much for your time. I also want to appreciate our special guest and our collaborators at Drosophila for their support through the years and their support to us thus far we have done. We say thank you. And finally, I want to thank the organizers of this seminar. Thank you for the awesome organization. I think we had less uh, uh, problems or technical problems now, so we are really improving. We appreciate all your efforts. And finally, I will thank our participants. Thank you for the questions. Were stimulating. And we hope that in our next seminar, you will all also be present. Thank you.
Thank you very much, Mrs. Inas, for information and probably the closing remark.
Okay, thank you very much. It has been a real pleasure. I hope that we can collaborate in the future, in the near future, and go there
Thank you very much.
see in Drosophila being implemented in Rwanda. Okay.
Thank you very much, Prof. I hope when we call you next, you will find time to attend to us.
Okay, sure. I will. It was a pleasure. Thank you.
Greetings from you. I hope
Thank
you can hear me well.
Yes,
I'm Miss
yes.
Inas. I will just give you a 2 minutes briefing, and I want to thank our esteemed guest and speaker for the fruitful presentation. That was very interesting. I will also thank our modulators and participants for their great time and cooperation. I just want to inform you, we will be having a next webinar that will be held on the 23rd of October. So be aware of that, and I hope we all can join and get fruitful information from that. Thank you for your time and presence and participation. Thank you so much.
Thank you, everyone, for making today's program a reality. Till we meet again, do have enjoy the rest of the day. Thank you. Bye-bye.
Bye. Thank you all.